Hi everyone, my name is Roya and this is my presentation on excitable tissues. Neurotransmitters are often referred to as the body's chemical messengers. They are the molecules used by the nervous system to transmit messages between neurons or from neurons to muscles. Communication between two neurons happens in the synaptic cleft that modulate ion channels, which results in hyperpolarization of the cell by opening chloride channels and allowing chloride to enter the cell. This action prevents the transmission of the action potential. As shown in Figure 1, GABA is an amino acid and the primary inhibitory neurotransmitter in the brain. The main function of GABA is the neuronal synapse is to bind to postsynaptic GABA receptors. Due to the prevention of firing of action potentials, GABA reduces the excitability of the cell. The role of GABA is to reduce the activity of the neurons as it is an inhibitory neurotransmitter. Activation of ionotropic and metatropic GABA receptors by GABA reduces the excitability of the cell, inhibiting the firing of the neuron. Valium is a benzodiazepine that is commonly prescribed to patients with generalized anxiety, panic disorders, and OCD. So how do benzodiazepines reduce neuronal excitability? Benzodiazepines enhance the effect of GABA and reduce neuronal excitability even further than GABA alone. This medication is usually given to patients who suffer from anxiety, OCD, and panic disorders. As shown in Figure 2, benzodiazepines reduce the conformational change in the GABA-A receptor by binding to the pocket created by A and Y subunits. This alteration, in turn, induces a conformational change in the GABA-A receptor's chloride channel that hyperpolarizes the cell and accounts for GABA's inhibitory effect throughout the central nervous system. So why is the combination of barbiturates and benzodiazepines dangerous? Well, barbiturates are CNS depressants which acts the same as GABA-A receptors. An example of this is alcohol. Valium is a type of benzodiazepine that reduces neural activity. If this medication is taken in combination with alcohol, neuronal activity can be reduced even further, which can lead to sedation, unconsciousness, or in worst cases, death. Both alcohol and Valium are central nervous system depressants, and both interact with chemicals in the brain that send signals to the lower blood pressure, slow heart rate, and a reduced body temperature. When combined, these depressant substances can compound the effects of each other, increasing potential short-term and long-term side effects. Chloride ions also play a role in skeletal muscle. So what is their function? Chloride channels are responsible for chloride ion movement in skeletal muscle. As shown in Figure 4, the ClC1 chloride is an ion channel which is responsible for 80% of the resting membrane conductance in inactive muscle fibers, which repolarizes action potentials to stabilize resting potential. The ClC1 can be further activated by depolarization to bring the membrane excitability back to resting state. Channelopathies are pathological conditions that affect ion channel function. An example of this is myotonia congenita, as shown in Figure 5, which is the foremost common skeletal muscle channelopathy and is caused by transformations within the skeletal muscle voltage-gated chloride channel gene. Ordinary muscles require a high resting chloride conductance for quick repolarization of the T tubules and stabilization of the electrical excitability of the muscle membrane. In myotonia congenita, these transformations cause a decrease in chloride current that results in repetitive depolarization of the muscle fibers. The muscles do not relax properly after voluntary contraction. Symptoms include difficulty swallowing, gagging, stiff movements, shortness of breath, and difficulty opening eyes after forcing them closed. Treatment for this condition include medicines such as mexiletine, phenytoin, and procainamide. Thank you very much for listening to my presentation.